Food, when eaten, begins to digest in the mouth, but different types of food are digested in different parts of the digestive system. Protein, while masticated in the mouth, is primarily digested in the stomach. Fats are primarily digested in the small intestine. The process of carbohydrate digestion begins in the mouth with the release of the amylase enzyme. In the mouth, amylase enzymes begin to break apart the bonds holding the carbohydrate polysaccharide sugars together. As masticated and partially digested food reaches the upper stomach, the fundus, through the esophagus, the lower stomach begins to fill with hydrochloric acid. Additional carbohydrate digestion continues from the mouth in the upper stomach. Protein is primarily digested in the stomach. Protein digesting enzymes break apart the bonds holding the amino acids of protein. Digested protein, partially digested carbohydrates, and undigested fats travel into the small intestine for the digestive process to be completed. In the small intestine, amylase completes the digestion of carbohydrates. Fats are digested in the small intestine by the lipase enzyme. Digested protein, carbohydrates, and fats are then absorbed through the villi of the small intestine into the bloodstream, where crucial life-sustaining nutrients are distributed throughout the body.
What's up, dudes, and what is up, beautiful ladies out there? Brian's sitting here enjoying an ice cream cookie sandwich, which is packed full of fat molecules, some protein molecules, and a ton of carbohydrate molecules. So how does our body digest and absorb the carbohydrate molecules from food? A quick review. There are three main types of carbohydrates. Monosaccharides, which are single molecules, disaccharides, which are two monosaccharide molecules bonded together, and polysaccharides, which are many monosaccharide molecules bonded together. And some shorter strands of polysaccharides are often called oligosaccharides. Our bodies produce and release enzymes that basically break down larger carbohydrate molecules into smaller units. There are two places that carbohydrate digestion or breakdown takes place: in the mouth and in the small intestine. In the mouth, salivary glands release salivary amylase, which is an enzyme that starts breaking down starch molecules. Starch is a polysaccharide molecule that contains a large number of glucose units bonded together, and it is found in many foods, including rice and potatoes. Once the food or bolus is swallowed and enters the stomach, the low pH of the stomach inactivates the salivary amylase. There's virtually no digestion of disaccharides until they reach the small intestine, and we'll cover them in a minute. As the bolus moves from the stomach into the duodenum and jejunum, which are the first parts of the small intestine, the pancreas releases pancreatic juice into the small intestine. This juice contains pancreatic amylase, which is an enzyme that continues the breakdown or digestion of starch into shorter strands of glucose. The brush border or microvilli, which are tiny hair-like projections located on the enterocyte cells of the small intestine, also contain enzymes that finish the breakdown of starch into single glucose molecules that are ready for absorption. Now for the two-unit disaccharides, which my ice cream cookie had a ton of. So the three main disaccharides are sucrose, or sugar, which is comprised of a glucose molecule and a fructose molecule, lactose, which is galactose and glucose, and maltose, which is two glucose units. The enzymes that act on these are sucrase, lactase, and maltase, which are also located on the brush border of the enterocyte cells. So basically the body has broken down these carbohydrate molecules into glucose, fructose, and galactose units. Glucose and galactose are mainly absorbed into the enterocyte cells through an SGLT1 protein transporter on the membrane of the cell, and fructose is absorbed into the cells through a GLUT5 transporter. On the other side of the enterocyte cells, glucose, galactose, and fructose are transported out of the cell through a GLUT2 transporter into the blood, where they travel to the liver. In the liver, fructose and galactose are mainly converted into glucose. The actual glucose molecules from food can be taken up by the liver and stored as liver glycogen, or they can become part of blood glucose and be used by cells throughout the body as energy. They can also be taken up by muscle cells and stored as muscle glycogen, or they can be converted into fat and stored in adipose or fat tissue. And folks, that be the basics on how carbohydrates are digested and absorbed. You can hit the thumbs up or thumbs down. If you have any comments, you can leave those down below. Till next time, folks, I am out of here. Oh, pretty good. It's protein time, baby. So how does the body digest and absorb all the protein molecules we get from food? Before we get to that, let's do a quick review of proteins. Without going in depth, proteins are large molecules made up of amino acids. Amino acids are linked together by peptide bonds to form a polypeptide chain. And some proteins are single polypeptide chains, and other proteins can have folds and many polypeptide chains linked together. There are two main places that protein digestion takes place: in the stomach and in the small intestine. Once the protein molecules are swallowed and enter the stomach, chief cells in the stomach release the enzyme pepsinogen. Pepsinogen is an inactive enzyme because if it were active in the cells, it would destroy proteins located inside the cells. Hydrochloric acid released inside the stomach causes a low pH level, and this actually activates pepsinogen to pepsin. And it also denatures the proteins or changes the shapes of proteins, causing them to unfold. Pepsin breaks down the proteins into smaller fragments. So next, the food or chyme enters into the small intestine from the stomach. The higher pH of the small intestine inhibits pepsin from further breakdown of the partially digested protein. However, there are three enzymes that are released by the pancreas into the small intestine that do continue digestion: trypsinogen, chymotrypsinogen, and procarboxypeptidase. All three of these are inactive when released, An enzyme called enteropeptidase is released by the small intestine, and this enzyme activates trypsinogen to trypsin, which in turn activates other trypsinogen into trypsin, and chymotrypsinogen to chymotrypsin, and procarboxypeptidase to carboxypeptidase. Trypsin and chymotrypsin act on bonds between specific amino acids within the partially digested protein, breaking them into smaller peptide fragments. Carboxypeptidase can only act on a specific end of a fragment, breaking the bond and releasing one amino acid at a time. So at this point there are free amino acids, dipeptides, which are two amino acids bonded together, and small peptide fragments. The brush border or microvilli, which are tiny hair-like projections located on the enterocyte cells of the small intestine, also contain enzymes that finish the breakdown process. The brush border enzyme dipeptidase breaks the bond between dipeptides, and aminopeptidase acts on a specific end of a fragment, releasing one amino acid at a time. Free amino acids are then absorbed into the enterocyte cells through membrane transporters within the membranes of the cells. Some di- and tripeptides are also absorbed into the enterocyte cells through specific membrane transporters, where they are then broken down into free amino acids. On the other side of the enterocyte cells, the amino acids are transported out of the cell into blood, where they then travel to the liver. Some of the amino acids will be taken up by liver cells, some will be used by cells throughout the body as building blocks of new proteins, and if there are excess amino acids, they can be converted to glucose, or they can be broken down and used as fuel for cellular respiration. Alright folks, so that be the basics on the digestion and absorption of proteins. If you have any comments, you can leave those down below. You can either thumbs up or thumbs down. Till next time, I am out of here.
What is digestion? Digestion is breaking down of particles by means of mastication or chewing and by the use of enzymes. What is absorption? Absorption is the transport of nutrients or substances from one place to another. It passes through the lumen to the blood vessels and undergo metabolism. Let us focus on the digestion and absorption of fats. Fats are macromolecules that are made up of lipids. First, fatty acids, which are subunits of lipids, are collected together, forming triglycerides. These triglycerides are further combined to form what we call lipids. Let's say a person ate pizza, which contains fat. It enters first the mouth, but the mouth does not contain bile. Therefore, no digestion happens and continues to descend, passing through the esophagus to the stomach. If you are younger, you can digest short-chain fatty acids because gastric lipase is quite stable in acid environment. As a person ages, stomach is very acidic and gastric lipase enzymes become inactivated. So nothing really happens in adult stomach and fat digestion. The maximal fat digestion process starts in the duodenum. The gallbladder secretes bile to emulsify lipids, and upon emulsification, the pancreas secretes the enzyme lipase to break down the triglycerides into monoglycerides and free fatty acids which are not soluble in water. Monoglycerides and free fatty acids cannot enter the cell, but with the aid of micelle from bile salts, monoglycerides can enter the cell. A typical micelle in aqueous solution forms an aggregate with the hydrophilic head regions of the microvilli present in the cell surface. Once inside the cell, the free fatty acid and monoglyceride enter the endoplasmic reticulum, which is a system inside the cell whose functions include synthesis and transport of lipids. Here the digestive products are resynthesized into triglycerides, which is the major form of fat stored by the body. The triglycerides is packaged along with cholesterol and fat-soluble vitamins into chylomicrons. They serve to transport dietary lipids through the body. They leave the mucosal cells via exocytosis and enter the lymphatic vessels in the submucosa. From here, they are transported to the thoracic duct where they enter the bloodstream.